Hello, welcome back. This is part 3 of our look at the urinary system. This is Professor Young. And before you is a series of terminologies that we need to be familiar with as we move forward, looking at exactly how the proximal tubule, the loop of Henle, and the distal tubule, as well as the collecting ducts, all work in creating this final byproduct that is of which is urine. Of the terms that are in front of you, some of them are obviously more essential to our understanding than others. And of those that we need to make sure that we understand is solvent drag, symport, antiport, as well as the sodium glucose transport proteins. These four terms are of most importance to us because it really describes to us how the proximal collecting ducts, the proximal collecting tubules, the loop of Henle, and the distal tubules really do what they do. And it all comes down to pressure gradients and concentration gradients, as well as some channels that are going to allow for the transportation, whether it's the use of energy or just by diffusion. As is the case with the solvent drag, water here is the key, and then on the next slide you'll see exactly what I'm talking about. But water is the key for acting as the solvent that is going to dissolve all of the solutes that are found within the tubule filtrate. And what will happen is that water will move from an area of higher pressure to an area lower concentration to an area of lower concentration, higher to lower. And so that water will move out of the proximal tubule as well as the loop of Henle and go back into those peritubular capillaries. And when it does that, it will bring with it a whole host of different solutes that we will see in a few minutes. Symport is another mechanism of transporting sodium and usually something else. In the case of, sometimes it'll transport calcium, sometimes it'll be chlorine, sometimes it'll be potassium, but typically as sodium leaves, something else will leave with it. And we'll see where that's going to take place again on the next slide. And then we have antiport, where one thing is coming in and the other thing is going out. And so in the case of looking at renal function, typically sodium is going to be reabsorbed back to the epithelial tissue from the tubule filtrate, and then hydrogen ions will move from within the epithelial tissue back and will move into the tubular filtrate. And then sodium glucose transport proteins, and this is basically glucose hitching a ride on sodium as it is being removed from, again, typically the filtrate into the epithelial tissue, crossing the extracellular fluid and moving back into those peritubular capillaries that we will talk about. Again, glucose is not supposed to be found within urine. And we'll talk more about that, but the bottom line here is if glucose is not completely reabsorbed, typically that is indication of diabetes within the person, and that'll become clearer as we get further along into this presentation. So these are the key terms that you need to be aware of. Again, solvent drag. Symport, antiport, and the SGLTs. Making reference to what I was just talking about as far as really being able to visually see and understand solvent drag, symport, antiport, and the SGLTs, we've got this diagram here, and I will happily point out to you exactly where things are. And so here is an example of a SGLT, a sodium glucose transporting protein. All right, so we can see that the sodium here is moving from the tubular filtrate. Here is the tubular filtrate, outlined here in yellow. All right, the epithelial tissue, again, is outlined here in blue. The interstitial fluid is outlined here in purple, and then outlined in red would be the capillaries. And what we're looking for here is that typical movement of moving from tubular filtrate back into potentially the peritubular capillaries. And when we do that, as will be discussed on the next slide, we simply refer to this as reabsorption. Because we are reabsorbing from the filtrate back into the capillaries, which is where the initial filtration occurred. If you remember, the afferent arterioles will enter into the glomerulus. It will filter out due to pressure gradients. It'll filter from the glomerulus into the Bowman's capsule, and then that arterial system will leave the glomerulus as the efferent arteriole. And to a certain extent, those efferent arterioles will help to make up the peritubular capillaries that are now winding down alongside of whether it is the proximal tubule, the loop of Henle, or the distal tubule, or even the collecting tubules or the collecting ducts. So here we have this SynPort mechanism going, and really that's all the sodium glucose transport protein is. It's an example of SynPort because it is transporting, again, sodium and something else. In this case, it is glucose. But both of them move, both of them are being reabsorbed back into first the epithelial tissue, then back into the peritubular capillaries. And you can see that right here. If we just follow glucose, we can see that glucose now ends up over here, and its final stage or resting place is going to again be back within the peritubular capillaries. Sodium kind of takes an interesting twist, and sodium is actually helped to be removed in a number of different ways. If we look over in this area, we can see that again as an antiport, sodium is being removed from the filtrate. At the same time, hydrogen ions are being brought back into the filtrate, and we're going to talk about that in a few seconds. But I want to follow sodium on its journey here because we see something very interesting begin to happen. Over within the epithelial tissue, we see this sodium-potassium pump. And what that sodium-potassium pump is doing is, first of all, it is using our good old friend's ATP. Oops, that was ugly. It is using our good old friend's ATP here to run this sodium-potassium pump. That's nothing new. We understand that. But the significance of this is, and this is where the criticalness comes in, as sodium is leaving, glucose is leaving on its own. It's going. It was pulled from the filtrate into the cells. Once it's in those epithelials, it's going to go right back into the capillaries. Sodium needs to be forced out. As sodium is being forced out, potassium is being pumped in. But look what happens to potassium. Potassium is being pumped in for the sake of releasing sodium, but at the same time, potassium is also being pumped back out. And what is referred to as the potassium chloride SynPort. Potassium and chloride are following sodium out. What does potassium chloride equal? Potassium chloride equals salt. What does sodium chloride equal? Sodium chloride equals salt. Why is that important? That is important because we're creating an environment within the capillaries that is leading to a higher concentration of salt, which is going to do what within that tubular filtrate? It is going to, whether it wants to or not, it is going to pull water. And it's going to pull the water from within the filtrate and reabsorb it back into the capillaries, because it's going to want to move from an area of higher solutes. It's going to want to move from an area of lower solute to an area of higher solute. Higher solute here being the case of the salt, whether it's potassium chloride or sodium chloride. And so that water is going to move. And when it moves, look at everything it's going to pull with it. Here's the concept of solvent drag coming into play. It's going to move urea. It's going to move uric acid. It's going to pull more sodium with it, more potassium with it, more chlorine with it. It's going to pull magnesium, calcium, and phosphate. It's going to pull it all with it. And so we see all of this work that was done within the glomerulus to filter all of this stuff out. But because water is so key to our survival, it's going to go ahead and it's going to reabsorb all of that water. In order to do that, it needs to set up concentration and pressure gradients. And it's a matter of fact that when water moves, because all of these things that we just looked at, urea, uric acid, sodium, potassium, chloride, magnesium, calcium, and phosphate, all of that is dissolvable in water. So as the water goes, so does all of this. Why does it seem like we're doing double the work? Well, it seems like we're doing double the work, and actually, let me just skip this. That's the middle part. The end result is going to be this. The end result is going to be this. At some point in time, we're going to need to do the opposite of reabsorption. And the opposite of reabsorption is secretion. All right? We've reabsorbed all of this stuff back into the capillaries. Now we're going to take a certain part of this and we're going to secrete it, and we're going to secrete it back across those epithelial cells back into the tubule filtrate, or back into the tubules themselves, whether it's proximal or loop of Henle or distal. But here's the key. Why do we do all of this extra work? Why are we filtering and reabsorbing and secreting all the same substance back and forth? If we got rid of it, get rid of it. It comes down to this. The kidneys are filtering 180 liters a day of filtrate. That is a buttload of fluid. That is a buttload of nutrients. That is a huge amount of waste. 180 liters is 90 2-liter soda bottles lined up one side by side by side by side. That is how much filtrate is being removed from within your blood plasma on a daily basis. And the nephrons, all 100.2 million nephrons inside of each kidney, 2.4 million nephrons between two kidneys, are taking 180 liters of filtrate per day, and they have to reabsorb it to the point where it gets it down to one soda bottle. Your body, your nephrons, and your kidneys, think about the marvelness of this. It's taking 92-liter soda bottles, and it's condensing it down to one. And if it didn't do that, you wouldn't be able to make it from your bedroom to your bathroom in time before you had to urinate. It wouldn't happen, let alone drive 40 minutes to go from your house to campus. And so it's vital that we seem like we are being redundant, but it's so vital when we look at the overall movement. Reabsorption continued here, now that I've come off of my exciting tirade that I just went on. We know it runs by osmosis. We know it runs by solvent drag. We know the end result of reabsorption is to get a certain amount of this material back into the paratubule capillaries. How do we do this? Well, it comes down to really the space in between the tubule and the capillaries, the interstitial fluid. And it has to maintain a high interstitial pressure to allow for pressure gradients to be able to move from to be able to flow the way that it needs to flow. Within that higher interstitial fluid, we need not forget COP, colloid osmotic pressure. The pressure and therefore the ability to conduct osmosis based on suspended solutes within the liquid. That is going to help dictate which way fluid is going to move. And so this is why you have all of those syn ports and antiports ATP pumps, those sodium potassium pumps, because what you're trying to do is create a higher COP out in the interstitial fluid, out in the peritubular capillaries. Because what that is going to do is it's going to set up a scenario where that water is going to want to move from inside of that filtrate tube, whether it's the proximal tubule or the loop of Henle, and move into the interstitial fluid and or the peritubular capillaries. And so this really becomes critical for us. The other thing that you need to look at here has to do with your diameter difference. Here's the diameter of the afferent arterial. Here's the diameter of the efferent arterial. Notice, and this is not just a mistake within the drawing. The diameter of the efferent is very much more narrower, and the reason why it does that is because it will increase blood hydrostatic pressure. It increases the pressure found within the capillaries. All right. And that plays a part in helping to control overall blood pressure throughout the body. Now, how do we know when enough is enough? How do we know when we have moved enough solute across in order to have an effective amount of water move with it? Well, it has to do something that we abbreviate TM, T subscript M. Transport maximum. And transport maximum works off of the philosophy of your nephrons have, and associated structures, the epithelial cells, the podocytes, everything like that, have a certain amount of transporters. They have a certain amount of syn ports, they have a certain amount of antiports, you have a certain amount of sodium potassium pumps and all that kind of good stuff. As long as you are under the number of transporters that you have or equal to, as long as you are less than or equal to, as long as you are less than or equal to the number of transporters versus the amount of solute that you have, everything will flow as it should. The problem comes in that each and every transporter has a transport max. In other words, it has a critical value at which it's going to say, I'm overwhelmed and I can't pump anymore. Some of this stuff is going to be left behind in the filtrate. And that occurs when the solute molecules are greater than the number of transporters. And when you have more solute molecules than what you do the number of transporters, it's inevitable. That solute is going to end up within the urine. And this is key, especially when we look at things such as glucose. Right? This is key, especially when we look at things like glucose. The Tmax, right, the transport maximum for glucose is 320 milligrams of glucose per minute being moved across from the filtrate into the epithelials back into the peritubular capillaries. The normal individual has enough transporters to move 320 milligrams per minute of glucose. We function normally at around 125 milligrams per minute. So that gives you a buffer of almost 200 milligrams per minute for glucose. In diabetics, they exceed that 320 milligram per minute. They exceed that buffer of 195 milligrams of glucose within the filtrate. They exceed it. They go beyond it. And therefore, the glucose ends up in the urine, and that's one of the key indicators that somebody is diabetic. When we talk about secretion, and then if you remember, I basically said that secretion was as we move from the paratubule capillary back into the filtrate, and that's exactly what we see happening here. We see tubular secretion when we go from the capillaries back into the tubular structure. And the reason why we're doing that is because look at what we're removing once again. Pretty much everything that the water removed through solvent drag, we now have to take back out. Uric acid, bile acids from within the digestive system, ammonia, NH4+, catecholamines, which we know are released from the adrenal gland. It's made up of dopamine. It's made up of epinephrine, norepinephrine, prostaglandins, which are active within the process of swelling. Right? Creatinine, which is the byproduct or the waste product, nitrogenous waste, mind you, of creatine phosphate, which is used to help to restore ADP. Secretion is also going to, if the blood is too acidic, it's going to remove hydrogen ions. If the blood is too base or alkaline, it's going to remove the bicarbonate ions. Right? But it doesn't stop. It doesn't stop there, because secretion is also going to remove. Secretion is also going to remove things like morphine from the body. Right? It's also going to remove things such as penicillin. If I can remember how to spell penicillin. There we go. It's going to remove things. It's going to remove things such as aspirin, which is why you have to take this stupid thing every 4 to 6 hours, because your body is removing it. It's got a high rate of clearance. It's got a high rate of clearance. All right. Other drugs, vitamins. Right? Vitamins is key here. You waste about 85 to 90% of your vitamins that you take. You waste about 75 to 80% of the drugs that you ingest into your body. They are cleared through the kidney system, urinated out. They're just wasted. Completely wasted. But if you understand pharmacology, the science of studying pharmaceutical medicines and how they impact on the body, what you understand is that you have to have such a high rate of waste to ensure proper. I hope I'm remembering this right. Proper efficacy. In other words, each medicine, each drug that you take and put in your body has a certain affinity for binding to receptors that are going to cause the desired outcome within the body. If you don't have enough of that drug in there to meet the efficacy of that drug for it to work effectively, you're wasting your time taking the drug. And so what pharmaceutical companies have done and what drug manufacturers have done is they've exceeded the efficacy rate within the medication to guarantee that before the urinary system clears that drug out, it stands a fighting chance to bind to those receptors so that you have the outcome that you need. And so there is a reason for why we waste so much. And if we had more time, we would get into this, but unfortunately, we don't. All right, distal convoluted tubules. All right, within the distal convoluted tubules, we see more reabsorption. We see more reabsorption. And again, the key things that I want you to know about here have to do with aldosterone. For heaven's sakes, young. Has to do with aldosterone right here. as well as antidiuretic hormone down there. Again, aldosterone is going to be released. It is a hormone. It's going to be released when blood pressure drops. And the reason why it's released when blood pressure drops is because we need to look at it from the perspective of blood pressure has dropped. Now, aldosterone will be released, and it will come in and it will reabsorb salt. In other words, it'll make the capillaries and the interstitial fluid, the extracellular fluid, retain salt. It'll pull it out of the filtrate. And as soon as you increase the level of salt within the capillaries, the peritubular capillaries or the extracellular fluid, what's going to follow with it? Water. So now you're pulling water out of the filtrate and you're pulling it back into the cardiovascular system, which we know will increase hydrostatic pressure. And when we increase hydrostatic pressure within the blood vessels, we increase blood pressure. And so aldosterone is a key hormone in doing that. Antidiuretic hormone. We're going to talk about the collecting tubules or the collecting ducts here in a few minutes. And antidiuretic hormone specifically acts on them, and it makes them more permeable to water so that the water is going to be removed from the filtrate and put back into the system again. Now, the collecting duct or the collecting tubule, as I will sometimes see, and that's what all these CTs are, it just stands for collecting tubule. The collecting ducts or the collecting tubules, the lower end is where all of the action happens because it becomes a hypertonic solution. In other words, you've got more water on the inside than what you do around the surrounding. And I'm going to talk about that here in a couple minutes. And that'll be crystal clear to you why this has to happen. But what it'll do is that it'll draw more water out, and that'll make the urine highly concentrated. So that way it'll go ahead and leave as it needs to. All right, 2 more slides and we'll be good to go. This is a slide here that I need you to focus in on because this here describes everything that you ever wanted to know and then some about urinary function. All right, so up here we have the proximal convoluted tubules. Down here we have the loop of Henle, and then over here we have the distal convoluted tubules. And so here's what's happening. Glomerulus filtrate, it enters into the proximal tubule. It flows through the proximal tubule where, notice, water is removed. Now, water is removed from this area because we've seen this already. If we go back to this slide, we know water is going to move because of everything that is happening within this region. All of those solutes are moving out. And when they move out, you're going to create a higher osmolarity. You're going to create a higher presence of solutes within the extracellular fluid than what you are within the tubule, and that's going to draw the water out. And that water collection, that water reabsorption, continues down the proximal tubule into the loop of Henle. On the other side of the loop of Henle, on this side, as we enter into the upper portion or the distal portion of the loop of Henle, heading into the distal convoluted tubules, notice what happens. We see a complete shift in what is being removed. On the other side of that loop of Henle, we now see salt. We now see sodium, potassium, and chlorine being removed. Sodium chloride is salt. Potassium chloride is salt. You are creating a highly concentrated, high osmolarity system within the renal medulla that is highly concentrated with salt, which is going to cause what to happen? It's going to cause more water to be removed. But it's not going to cause more water to be removed on the upswing of that loop of Henle. It's going to cause that water to be removed, wait for it, over here in the proximal collecting tubule going down into that beginning or that ascending limb of the loop of Henle. Remember what we said. When that loop of Henle enters down into the medulla, whether it's a juxtamedullary nephron or whether it is a cortical nephron, the loop of Henle always dips down into the renal medulla. And remember I said there is higher pressure down in that renal medulla, and that is going to add to water reabsorption. Bam! Here it is. That salt is being removed from the distal collecting the distal collecting tubule and the distal portion of the loop of Henle, and it's that salt is going back into the renal medulla, where it's creating the higher pressure, so that as the filtrate enters into the proximal tubule, into the ascending branch of the loop of Henle, that water has no chance in staying in that filtrate. It's got to leave. It gets the double whammy. Not only is this whole area working to make sure that the water leaves, but now in the distal collecting tubule, we have all the salt leaving, which is going to increase the pressure. We know that water is going to move from an area of lower solute concentration to an area of higher solute concentration. There it is. This picture here summarizes everything. I'm going to let you look at this for about 30 seconds. As you are looking at this, study this, know where we are going. Know where filtration is happening. Know where reabsorption is happening. Know where reabsorption is happening. I totally messed up reabsorption, but you get the point. Know where secretion is happening. Know it. Right? Know it. The last thing I will say is bada bing, bada bam, there's your collecting tubule. There's your collecting duct. Right? And we know that multiple nephrons are going to end into this. I end this session the way I end all my sessions. Go back, review any sessions or any spots of this that you need to look at. Mute me out if you just want to focus in on the diagrams. Mute me out and pause it. If you have any questions, shoot me an email. I'll be more than happy to answer them. Otherwise, write them down and ask me during the next lecture. And this is Professor Young saying happy studying, and we'll see you soon.